excited to be participating in in-person meeting again. And thanks to the organizers for making this happen. So I'm Manolis Maragkakis. I'm a Stanman investigator at the NIH, specifically in the National Institute on Aging. So today I would like to talk to you a little bit about some of the latest work that we're doing in my lab to capture the state of the RNA using direct RNA sequencing to discover changes upon different conditions. In this case, we're focusing a lot on cellular stress. So as you probably everyone here knows, so the mRNAs, the structure of the mRNA is pretty well defined. This is how we think about RNA. So RNA has a 5' cap in the beginning, and then has 5' UTR coding sequence, 3' UTR and polytails. And then ribosomes are recruited and translation initiates until we have mature protein product. And at some point, these RNAs are exiting the translational pool, and we have first the adenylation, which is considered the rate limiting step, and then there is fragmentation happening from the 5' end. And these two processes were mostly considered distinct processes. Now, a few years back, what we tried to do was to try to capture, we were very interested in capturing the RNA state, and like in what is the exact state that RNA exists in cells, right? So we're very focused to see how these, the end, the 5' and the 3' ends of the RNAs are being defined and how they might be regulated. So I don't see the left part of my slides here, but so what we did essentially was to develop two different protocols, which we call ACRON 3 and ACRON 5. And essentially what we're doing in the ACRON 3 and ACRON 5 is that for the ACRON 3, we isolated capped molecules, capped mRNAs, and then we sequenced with short-read sequencing their 3' ends. And in the ACRON 5, we did exactly the opposite. We isolated polyadenylated RNAs, and then we sequenced with short-reads their 5' ends. Now, if everything was according to the textbook view of the mRNA life cycle, we would expect that the majority of mRNAs in cells would be mostly full length, right? That's what we expected to see. So if mRNAs were mostly full length, we'd expect, for example, to see their 3' ends that we capture. We would expect to see almost all of them at the 3' end of the annotated transcripts. Same for the 5' end. But that's not what we see. We actually see that many of these ends that we capture actually fall within the mRNA body. And this essentially changes the way we think about RNA. And this shows that probably in cells, RNA is in a different state than the full-length version of it. So, but most importantly, I think in this work, what we found is that these areas where we see these RNAs occurring, where these caps are happening on the RNA, are actually very conserved. They're much more conserved than the surrounding areas. And that indicated to us that these sites are not probably randomly distributed on the mRNA, but probably they're very heavily regulated. And when we see regulation, we can obviously think of deregulation, particularly in the context of diseases. So, but one of the main problems that we had with these two protocols was that there were essentially two distinct protocols. There were two different protocols. And we couldn't really quantify, first of all, the interaction between the 3' and the 5' end. So we didn't know if one has an effect on the other. But also we couldn't quantify how much fragmentation we have from the 5' and how much fragmentation we might be having from the 3' end of mRNA molecules. So to address this question, we turned to nanopore sequencing, and we heard a lot about nanopore sequencing and long read, so I'm not going to go into too many details here. The only thing I'm going to say is that the DNA and the RNA translocates through a protein pore, and essentially the device measures the electrical current. And the fluctuation in the electrical current at different bases passed through the pore is what defines essentially the base calling step. And the benefit of the technology, obviously, is that instead of being able to sequence a few hundreds of nucleotides, we can sequence much longer molecules. So our hope was that we would be able to capture full-length molecules using this technology. Now, as you might know, when we are doing direct RNA sequencing, the sequencing actually proceeds from the 3' to the 5' direction. And one of the major limitations in nanopore sequencing is sometimes it doesn't reach all the way to the 5' end, right? So to address this problem, what we decided to do was to ligate, actually, before we even do any library preparation, we ligate a 5' and adapter at the 5' end of the molecules. And what that does is essentially ask us a marker inside our sequencing data to be able to know that if we actually see this sequence in our sequencing data, we know that we reached all the way to the 5' end and we capture the 5' end. But very importantly, this also allows us to play a little bit with some of the pre-processing step on the RNA, and therefore mark different kinds of RNA molecules. So I'm not going to go into details here because this is part of a published work that we recently published. But what I want to focus is that one of the treatments that we did is we first did a CYP treatment to remove 5' phosphorylated molecules, so molecules that in the state of being degraded. And then we did a decapping step to remove the cap from the mRNA molecules. And this allowed us to ligate the adapter on the capped molecules. So these molecules that we're capturing here have a poly A tail because that's how we capture them on the 3' end, and they also had a 5' cap. So this should be essentially the full-length molecules in the RNA. So one of the first analyses that we did is to try to see how these data that we capture, how these 5' ends, full-length molecules that we capture, map compared to what we have from all the annotations that are out there. For example, ensemble, UCSC, so on and so forth. And to our surprise, what we saw is that actually very, very few of the actual transcripts align together with where the annotated 5' ends are. And for the majority of the transcripts, we see that there is a great discrepancy between where the data is telling us the 5' ends of these RNAs are and where the annotation is actually telling us where these data are. Here, I just have one extreme case where you see that the annotation is over here on the left-hand side, and actually the data point us that the actual transcription start site is probably more than 500 nucleotides downstream of where the annotated end is. And there are only few mRNA molecules that actually go all the way to the 5' end. And annotation tend to be a little bit sensitive on getting the furthest away that they see sequencing reach probably. But that doesn't represent what is the actual state of the RNA in cells, right? So in this work, we also developed a technique to remove this discrepancy, essentially focusing on a data-driven annotation, and we re-annotated. Essentially, this work was done in HeLa, so we re-annotated the HeLa transcriptome, and we heard from Kim-Fa also, how there is a direction now where we have to think about transcriptome annotation in a cell type specific and perhaps tissue specific manner. And we also see that when we do this reannotation, this also aligns much nicer with cage data, which are supposed to be capturing the transcription start sites. Now, this was all published work, but the reason that we're doing all this work is because we're interested to capture the state of RNA under different conditions. And being in the National Institute on Aging, we're very much interested. 20 minutes, right? Okay, I'm like, okay, we're just starting. Okay, so we're very much interested in oxidative stress. And the reason that we're very much interested in oxidative stress, being in the National Institute on Aging, is because RNA oxidation associated with human aging and aging-associated disease, particularly neurodegeneration, such as ALS and Alzheimer's. So what Sokar and Silo and my group decided to do is to do a very simple experiment where they get control cells, and then they get cells that they treat with arsenite to induce oxidative stress. And then they perform the direct RNA sequencing with the ligate adapters to capture the RNAs. And here, first thing that we did is just a volcano plot, nothing special about it, differential expression, because we have the data. So all we did was just verify that the markers for the stress response are being upregulated, and there's nothing, that pretty much validates that the experiment worked. But now that we have this data at the single nucleotide resolution, we can dive in and look at individual mRNA molecules. And what we saw is, to our surprise, we saw that upon oxidative stress, we see massive shortening of the RNAs in the cells. So we see that cells are becoming, RNAs are becoming much shorter than the control condition. And when we look at only reads that have the 5' adapter, making certain that we actually reached all the way to the 5' and nothing funny happened in the sequencer itself or during the library prep, we see exactly the same thing. We see that upon stress, RNAs are becoming shorter. Now, when we go at the meta-length level to see which side of the RNA might be implicated the most, we see that the 5' end is much more heterogeneous than what the 3' end is, right? So the shortening that we're capturing, at least, is mostly mostly has to do with what is happening at the 5' end of the RNAs. Now, we see this shortening occurring, but we don't have a good statistical way to measure that. So we developed Nanoplan, which is essentially using linear mixed models, and it's the same concept as we would do a differential expression analysis, but in this case, we're doing a differential RNA length analysis. So the first thing was to, of course, do simulation. So we simulated reads at different expression levels per transcript and different shortening proportion. And what I consider one of the most important things is that under the null hypothesis, pretty much the software doesn't control pretty well the false positive rate. And that was important for us because we wanted to be specific and we didn't want to capture RNAs that are not being shortened. And now we apply this tool into the actual data. And quick validation, sanity check that indeed with the genes that the software reports as target for this stress-induced decay are indeed the ones that are showing the shortest lengths upon oxidative stress. But very importantly, we want to validate that in a completely different way. So we turn to QPCR experiments. But in this case, we used primers for different positions along in the mRNA molecule, right? We started from the 3' end, and we designed primers all the way to the 5' end. And what we see is that a very clear shortening as we go towards the 5' end. That's also a quick note for QPCR, but we probably already know that, how they're not really accurate for quantifying expression. Because if you design your primer at the 3' end of the molecule, you're probably going to find no significant change for this gene upon the two conditions. But if you design your primer for this area here, you're going to find a pretty significant change. So just a quick note, and we validate many of the top targets that we are identifying with Nanoplan this way. Now, having this data, we also want to dive into the molecular players that might be involved in this process. So we thought about XRN1, which is the primary 5' to 3' exon nuclease in cells. So we knock down XRN1, and indeed we see that when we silence XRN1, we see an increase of the RNA length. And most importantly, we see that whereas the genes that are not reported as targets by Nanoplan show pretty much the same length across the two conditions, when we actually look at the ones that are reported as the most as targets of the stress-induced decay, we see that these are the ones that are showing the biggest recovery in their length. Now, as I told you in the beginning, the traditional view for RNA decay posits that the first and rate-limiting step for RNA decay occurring is the adenylation. However, when we looked at our data, and we can do that because we're capturing both ends on the same molecule at the same time, what we found is that that's not actually the case. We don't see a difference in the poly-A length that affects 5' end decay. If anything, we're actually seeing a slight increase in the poly-A length, which is completely the opposite of what we would expect under normal decay conditions. Now, diving in a little bit more into some of the features of these target mRNAs for decay, what we noticed is that these ones have a small but significant difference in the coding sequence length. So we hypothesized that maybe this decay that is happening might have something to do with translation. And as you probably know, under stress, one of the first things that happens is that there is a phosphorylation of the initiation factor, EIF2 alpha. And when this factor is phosphorylated, we have a global translation initiation inhibition upon stress. So ribosomes are no longer recruited on mRNAs, right? So what we did is we tried to use chemicals. In this case, we used ISRIB, which is an inhibitor of the integrated stress response, to restore translation, so translation initiation. And we also use cyclohexamide to freeze ribosomes on the mRNAs upon stress. And what we see is that in both cases, we can pretty much rescue the length of these shortened RNAs, right? These RNAs are recovering the length back to their initial lengths, which indicates that this stress-induced decay is very much coupled to translation, and most importantly, it's coupled to the presence of the ribosomes on the mRNAs, right? So if you leave the mRNAs naked, then you have stress-induced decay. Now, coming into perhaps one of the most interesting plots that we have is we're seeing this 5' end decay initiating, right? So the expectation would be that these RNAs would eventually be cleared from the cells. So we would expect that we would see an association between decay occurring at the 5' end and RNA abundance in general. However, when we looked at it, we don't see any association. We don't see a correlation, and we don't see any difference on the differential expression between genes that are targeted for decay and genes that are not targeted for decay. And that to us meant that although decay initiates, for some reason, these RNAs that are being fragmented persist in cells. So they remain in cells, they're not immediately cleared. So that brought us to hypothesize that maybe this has something to do with the function of stress granules. And stress granules are membraneless organelles that consist of RNAs and RNA-binding proteins that are formed upon stress conditions. Here, we just visualize the formation of the stress granules upon the RNA treatment. And as previously reported by Ray Parker's group, these RNAs, the RNAs that are enriched in the stress granules are essentially generally longer RNAs that are enriched in the stress granules. But what we found is that these RNAs that are being enriched in the stress granules are the ones that are showing the greatest difference in the mRNA length, and they're being shortened the most. So the RNAs that are being shortened are essentially transported to the stress granules for storage. And here I'm finishing up. Okay, 3 minutes. Yes. 2 minutes. Yes, that's the last one. So, this is essentially our model and what I told you today, and that is that cellular stress induces widespread decay of mRNAs. The decay is independent of mRNA poly-A tail length, but depends on XRN1, and that decay is coupled to translation initiation inhibition and subsequent ribosome runoff. But most importantly, also these RNAs persist in cells, so they remain in the cells, although they have initiated decay, and perhaps stress granules are the sites where these RNAs are being localized. And our hypothesis is that this is happening because these cells that are under stress don't want to deal with decaying RNAs at that time. So they want to stop translating these RNAs, they stop translation, they start degrading these RNAs, but they don't want to deal with the massive amount of nucleotides that would be coming out from these RNAs, and therefore they store them to deal with them a little bit later. So with that, I would like to thank, of course, my team and my collaborators, and thank you for your attention, and I'll be happy to answer any questions.
Thanks, and great talk. I actually had 2 short questions for you. The first of which is, how do you control for arsenite incorporation into nascent mRNA strands? Because that will also cause for spontaneous RNA degradation. And then also, for your cap sequencing that you had, since there's several different types of 5' caps, is there a preference for one type of cap over another that you're seeing, for which is also in your data at the end there?
So I'll just describe it for. Yes, this one. So besides arsenite, we actually used hydrogen peroxide, and we see exactly the same thing. So it's not arsenite itself that it's doing that. But interestingly, we also mined data for heat shock. And we actually also see exactly the same thing. So it's not something that's happening on the RNA, like scission or something happening on the RNA. It's actually part of the stress response of the cell. Now, regarding the second question about the different cap type, I don't think we can distinguish at the moment between the different cap sites. So, but that would be very interesting to do in the future.
Other questions?
Thank you. Very interesting talk. I have two questions. One is more technology related. What we have observed is that direct RNA reads tend to be shorter than other long reads technologies. So do you think this is somehow affecting the type of results that you are getting? And the second question is, well, I can imagine that when you have like an oxidative stress that you kind of treat your cells, the cells are going to be stressed, right? And then you may have these kind of responses, but somehow you want to link this with the aging. So have you seen similar type of patterns in aged individuals?
Yeah, that's a great question. So the first question was why they are a little bit shorter, perhaps, than other techniques. Yes, it could be affecting, and that's essentially the reason that we went through the process of ligating these 5' adapters, so we can actually make sure that we reach all the way to the 5' end. I can't exclude that there still might be an effect from, from direct RNA sequencing having a little bit shorter lengths than other technologies. My feeling is that perhaps direct RNA-seq is a better representation of what is actually happening in the cells. But still, we need more data to quantify that. Now, on the second part, one of the important things that is here is that in order to capture this difference in the RNA length, you have to be very careful about the timing of your experiment, because if you leave these cells long enough, eventually these RNAs are going to be cleared out. So in cases where we're doing, for example, when we think about aging as a process, I would expect that at steady state, you probably are not going to see differences such as this one, right? So you probably need to monitor a transient state in order to be able to see this kind of effects. Does it make sense? So if the RNAs are generally shorter, yes, you could see it. But if it's a regulatory process where it's like something happens and these RNAs are starting to are getting shorter, then you have to make sure that you're not at steady state, because if you reach steady state, these are probably going to be cleared out and they're not going to be part of the sequencing. So I personally think that a lot of these things are happening in aging, and we're like doing the experiments now, and we have some preliminary results on that. But we need to be careful about the timing of the experiments.